Okay, I think I'm gonna start it off. I'm gonna share my screen and sorry. Okay, everyone can hear me, right? And if you have any questions, like just feel free to speak up or like interrupt me. I can't see my chat like the way that my computer is. So yeah, so either ask them in the chat or just speak up. But if you ask in the chat, I'll respond like afterwards. So I'm going to be presenting mainly about just some characteristics of the double helix, and all the information I'm using will be again from the lecture notes and from the molecules of life. And I'll post my slides after session. Okay. So we're going to talk first about sequence-specific recognition. So in replication and transcription, the DNA double helix has to be unzipped and opened, and then the material within can be read out like nucleotide by nucleotide, so that you get like an exact copy of the sequence. But the DNA helix doesn't necessarily need to be opened in order to gain information from it or to read it. Instead, proteins can read the base sequence from the outside of DNA via the grooves, usually the major groove. And so why is this necessary? Does anyone have any like ideas of why this would be important? You can like answer or not. Okay, so basically, this is directly from the textbook, but in order for RNA polymerase to initiate transcription, it needs to first be recruited to promoter sites on the DNA by proteins that bind to specific regions and then interact with the RNA polymerase. So the RNA polymerase needs to know where to bind, basically, and so it needs to be able to recognize sections of DNA without having already like looked at the DNA. And so kind of in simpler terms, there's proteins which are called transcription factors, which recognize target sites on DNA and then recruit RNA polymerase to synthesize the new chain, which can eventually lead to protein expression. And how can transcription factors recognize the correct target sites? They interact with the edges or grooves of the base pairs in dsDNA. And obviously this is like a little bit of a simplification. If you're really interested in this, MCB 110 like goes over transcription in detail, so you'll get more information there. But so again, back to the generals, there's 4 potential types of interactions with complementary base pairs, or like interactions that the transcription factor can have with the bases. Does anyone know any of these interactions? They're probably like more simple than you think. Okay, so if not, they're hydrogen bond donors, hydrogen bond acceptors, hydrophobic groups, and nonpolar hydrogen atoms. So like those are the various groups on the 4 bases or 5 bases that can be interacted with. So here's an AT pair. As you can see, I have the major groove and the minor groove noted, but so basically this first nitrogen at 1 is a hydrogen bond acceptor, and then the next one is a donor acceptor again, and then a methyl group or a hydrophobic group. So, and then down here, so you can see like the different colors delineate what kind of interaction it can have. But why is this important? So the pattern of potential contact sites forms the basis of a DNA recognition mechanism in which the protein positions complementary functional groups at the recognition sites. So in the major groove, these patterns of recognition elements are unique for each of the 4 base pairs, GC, CG, AT, and TA. And so you can distinguish which base pair it is by looking at these interactions. So you can tell if it's a GC, CG, AT, or TA. In the minor groove, that's not correct. You can distinguish between AT and CG, but AT is indistinguishable from TA and GC is indistinguishable from CG. So this is kind of like an image of that. You can see how the major grooves, they're like the pattern or order is different between GC and CG. but when you look at the minor group, it looks exactly the same. So if a protein was interacting in a minor group, it wouldn't be able to differentiate between a GC and CG by like just this means. And then this is just like a picture kind of showing, this is, I'm not sure what protein, but some protein interacting in the major groove of DNA, maybe reading it out. So that's for DNA, but the RNA double helix is a little bit different because RNA can't exist in B form because it doesn't adopt the C2 prime endo sugar pucker because of the extra OH group and like steric interference. And so instead, it forms an A form double helix or with the C3 prime endo sugar pucker. RNA DNA hybrids can also exist in the A form, and DNA can also exist in A form, but it's not as common. DNA will usually be found in B form. And so an A form it's different, but it's not like totally different. There are some similarities between A form and B form DNA, namely 2 antiparallel strands. It's a right-handed spiral. The phosphate groups are still on the outside with the bases inside, and there's a relatively similar number of base pairs per turn, but it's not exactly the same. I think it's 10 and 11. And then here's a picture of B form versus A form. As you can see, like the B form is like the typical picture that you would imagine, like when you think of DNA, and A form is a little bit different. It's good to be able to kind of recognize these two forms and also a third form, which we're going to talk about later, because on like an exam or later on in your coursework, you might need to distinguish just by looking at like visuals. So that's important. So one of the those were some of the similarities, but one of the most important differences between A form and B form DNA or RNA is the position of the base pairs with respect to the helical axis. So in bDNA, the base pairs are parallel to each other and perpendicular to the axis of the helix. Whereas in A form helices, the bases are tilted from the perpendicular and moved away from the center of the helix. This is kind of like hard to think about. It's better to like look at pictures and kind of spend some time visualizing it so that you can understand like what's going on. But this one's not labeled, but this is A form DNA and this is B DNA, and like the dot is like the axis. It's like looking down the axis. So yeah, those are like two pictures. They're both in the textbook that you can look at. And so ultimately, the A form has a deeper, more narrow major groove and a wider, shallower minor groove. This is because of the different positioning that we talked about in the last slide. And so the narrow, deep minor groove in major groove, in A form helices makes it more difficult for proteins to read out sequence-specific information at the edges of bases because they don't have access, like because of the shape. And so one of the main advantages of storing genetic information in DNA, not only is it more chemically stable, but it's much easier to control genetic processes via the readout of sequence-specific information in the major groove of DNA because it's accessible to transcription factors, or more accessible. There's also a third unique type of double helix called Z-DNA, which is like much different from A and B form. It's a left-handed helix, so it goes like the other way. Again, it will help to like look at pictures of it in order to kind of understand what it looks like and understand why it's that way. And so it's adopted preferentially by segments of DNA that have alternating C and G nucleotides. And since of Watts and Crick, it'll be on both strands. There's 12 base pairs per turn. The sugars alternate between C2 prime endo and C3 prime endo pucker, and G residues are in the SYN conformation while the C residues are in the normal anti-conformation. And so then this is I'm not really going to talk about it much, but it just kind of is a really good summary of the three different helixes that we need to know. And I'm not sure exactly like what you need to memorize, but just like kind of understanding the differences, especially the way they look and like right-handedness versus left-handedness, and why B form DNA like has its advantages is important. And then here's just an image of the three, and you can kind of see that A form DNA is the widest, and Z is not so wide. And then, so continuing on about the DNA double helix, it's very deformable. So as you can see from the images before this, it can be found in like many different forms and structures and still be in a double helix. The most common is B form, the traditional one, but this is not always true. Many local regions of DNA can deviate from B form while still maintaining Watson-Crick interactions and the double helical conformation. We can see this if we look at small molecules which bind to DNA and deform DNA away from B conformation as they do so. This is just one example. It's called like an intercalator. I'm not exactly sure it's pronounced, but it consists of aromatic regions which slip between the bases of DNA and cause local unwinding. So it changes the structure. And then there's a lot of other similar examples in the textbook if you're more curious about that. And then this is just one of the measures of deformability, which is persistence length, or the characterization of the stiffness of DNA. For B-form DNA, it's about 500 angstroms or 15 helical turns, and DNA shorter than persistence length is pretty rigid, whereas longer segments can bend without a large energy penalty. Okay, and that's my presentation. Does anyone have any questions on the slides or anything? Okay, if there's no questions, then the next tutor can start. You just send them in the chat too.
Okay, so let me just share my screen. Okay, so first today I'll be introducing the first part of DNA supercoiling. So I want to start talking about supercoiling by first reviewing some of the forces that contribute to double helix stability, because ultimately supercoiling is a way for the DNA double helix to relieve any sort of strain that it experiences. So I think it's good to understand, what forces are causing that strain in the first place. So if you remember from last week, I kind of showed you this diagram that I think is a good depiction of what base stacking looks like. And base stacking is one of the main drivers of DNA double helix stability. And the reason for that is because when you have bases like this, so in this case, cytosine and guanine, when they're stacked on each other and offset by an angle, then you can have regions that are relatively electron rich overlapping with regions that are relatively electron poor. And so then you have both dipole-dipole and electrostatic interactions that are contributing to a structure like this. And furthermore, it's because of these electrostatic interactions and dipole-dipole interactions that there's this 36-degree rotation between bases that's enforced. Because if you move away from this optimum 36 degrees, it's kind of like this energy diagram. This is just a generic energy diagram that many of you have already seen before. Because if you change the rotation of one of these bases and then you miss out on some of the optimum rotation, then you might have higher potential energy. However, if you start moving in one of these directions, then you could result in like more repulsion if you happen to overlap both electron positive regions or electron negative regions. So for this reason, the 35 to 36 degrees is kind of the optimum angle between bases as far as rotation goes. So the next thing I want to go over is kind of an example calculation to show how strain can be introduced to DNA double helix. So let's say, for example, you have a DNA molecule that's about 100 base pairs long. And because of this, it has 3,600 degrees of rotation. Because remember, if there's 36 degrees in between each base, then as each base is rotating, your DNA molecule is essentially able to accomplish 10 complete rotations, or 3,600 degrees along its length. And also keep in mind that DNA is right-handed, so these are all right-handed rotations. So now, if you were to apply a left-handed rotation, which is kind of going against all of the rotation that your molecule has accomplished so far, that's like subtracting 360 degrees from the molecule. And so this means that this negative 360 degrees would have to be redistributed among all the bases along that molecule. And so as a result of that, if you consider these two bases that were stacked initially, if instead the angle between them were, say, 32 degrees, then these two regions, which are relatively electropositive, could end up being closer to each other, and that could induce more repulsion. And this is the reason why, and the main takeaway from this is that the reason why DNA supercoiling happens in the first place is because adding strain to the DNA double helix and trying to force the bases to be in a different angle of rotation from each other can introduce a lot more strain. And it's just oftentimes a lot easier to bend the molecule as a whole because, as was mentioned previously, the DNA molecule is pretty deformable. And so next, before I get into sort of the solenoid form and the plectonemic form, I want to just review really quickly like difference between right and left-handed, because this was something that I found pretty confusing at the beginning of the course. So here's an image from your lecture slides, and this is the DNA molecule. So DNA is considered right-handed. And the reason for that is because of the way that it's drawn here. So when I look at a diagram like this and I want to determine whether this is right or left-handed, sort of the way I'd like to visualize this is if I try to picture this as a staircase and I try to imagine all the arrows as kind of steps on the staircase and the red line as the handrail on the staircase, I kind of ask myself, if I were to walk up this set of stairs, which hand would I use to grip the handrail as I'm walking? And so if I'm walking up, I think I would use my right hand to grip the handrail, and that's why this is a right-handed molecule. Another way of thinking about it is if you were to just sort of trace the red line with your hand, could you accomplish that by using your right hand or your left hand? And in this case, it's easier to do this with your right hand. And so this just kind of requires a little bit of practice. So hopefully by looking at images, you might get a better sense of this. Any questions so far? Okay, so next I'll move on to the plectonemic form, which is what happens when you have DNA supercoiling in circular molecules. So let's go back to the idea of what happens when you have underwound DNA. So when you have underwound DNA, the difference in the angles between the two bases is typically less than 36 degrees. And this is because, left-handed turns have been introduced. And this contrasts with overwound DNA, in which you have the angle between the bases being more than 36 degrees as a result of extra right-handed turns being introduced. So what does this look like if you had a circular piece of DNA? So if you can imagine this, and so I want to do just like a quick demonstration of kind of from just wires that I found around the house. Hopefully this is helpful. So if you imagine a circular piece of DNA like this, if you connect the strands, right before connecting the strands, if you apply some left-handed turns, which would be moving this strand like this. So if I were to apply these left-handed turns and then reconnect the molecule, what I then have is DNA that looks like this. And I think this might be a little bit difficult to view or resume, but hopefully this region where the color contrast is a little bit easier to see, you have the white part pretty much going over or crossing over the green part, and that's what's contributing to a right-handed crossing, and that contributes to negative supercooling, which I'll get to in a little bit later. And the same opposite scenario can be seen if you're doing overwound DNA, in which case you pretty much just have the opposite rotation. And as a result of that, the crossing in the plectonemic form will be will be left-handed, which will be positive supercoiling. So any questions so far?
Hi, yeah, I have a question.
Mm-hmm.
I'm not sure which one you said, but is it correct that underwound DNA has more like degrees of separation in between bases, and then overwound DNA has fewer degrees of rotation between bases?
So overwound DNA should have a greater separation than 36, and underwound should have a less separation than 36.
Okay, because in the overwound, maybe I'm looking at the picture wrong, but it shows like the 35 degrees, but then it also shows it going back a little bit, so I thought that the angle would be less, whereas for the underwound, it seems like the angle is more.
Right, okay, I think, okay, this might be a mistake in the diagram, but underwound should be more than 36. I'm sorry, underwound should be less than 36, and overwound should be more than 36. Okay.
Okay, sounds good.
Any other questions? Okay. And so the next thing I want to talk about is sort of the conversion between the plectonemic form and the solenoid form of DNA. So this is a diagram from your textbook. I think it's figure 2.226. And here you see an example of negative supercoiling. And here what's interesting to note is that the crossing in the plectonemic form is actually right-handed, because the strand is going over the one behind it. And if you follow it with your right hand, it kind of fits the curvature of sort of like the right-handed railing that I discussed earlier. And then if you just kind of apply these transformations to get it back to the solenoid form, then what happens is when you get into this sort of pretzel-shaped solenoid form, now it's actually left-handed. And for that reason, this is considered negative supercoiling because the sign that's associated with supercoiling is dependent, or it's defined based on what the solenoid form is. So this is pretty similar to something that was shown in lecture, in which you just had sort of the opposite scenario. So this is an example in which you have a solenoid form which was positively supercoiled because the solenoid form, these crossings were right-handed. But then if you convert it to the plectonemic form, and all of a sudden you have left-handed crossings. And so even though you might think left-handed, shouldn't that mean negative supercoiling? Well, actually, negative supercoiling, if it's left-handed, is only the case if you're looking at the solenoid form. So this is something that is pretty important to just keep straight. So I guess a quick rule of thumb is whatever the handedness is of the crossing in the plectonemic form, it's essentially the opposite when you're converting back to the solenoid form, meaning that if you have left-handed crossings in the plectonemic form, then when you're going to the solenoid, it's right-handed. And if you had a right-handed crossing here, then going here, you'd end up with a left-handed solenoid. And that's what defines either positive or negative supercoiling. And so next, so the next thing I'll talk about is writhe, and this is sort of an introduction to what happens in eukaryotic DNA. So far, all the plectonemic form, solenoid form, that stuff applies to circular DNA molecules. But what happens when you have linear DNA, such as like the kind of DNA that we have? And so in this case, it's important to recognize that a lot of DNA in eukaryotic systems is typically still fixed because there are lots of proteins that are bound to it. So for instance, your DNA has a lot of nuclear proteins that are attached to it that kind of anchor it to the nuclear membrane. So for that reason, it's still kind of kept under tension, although it's not in a circular form anymore. And so in these cases, it can still experience some strain if there are any like left-handed or right-handed turns that are applied to it. And so one way to visualize this is if you have like a spare pair of wired headphones, then you can essentially just hold the two ends fixed. And in this case, what's happening is that there is a left-handed turn that's being applied. So here, I'll use my left hand, and I'll apply a turn. And you should see that this is the type of crossing that you get. And if, kind of hard to switch between my perspective and your perspective, but this should look like a left-handed crossing because this strand is coming in front of the one right behind it. And that's why it's kind of a left-handed curvature. And so as a result of applying a left-handed turn to this DNA, you end up with something that's called writhe. And in this case, the writhe is negative because the crossing that you see here is left-handed. And this is something that we'll go over more in the second part of supercoiling that's coming right up after this. But really quickly, I want to go over the question that I put in the worksheet. So this is pretty much like verbatim from the worksheet. So if you want to reference it, I posted the link to Piazza right in the chat. But the question here is, if you look at this diagram of a version of plectonemic DNA, is this left-handed supercoiling, or is this left-handed or right-handed, first of all, and then does this imply positive or negative supercoiling? So does anyone want to take a guess at this? Okay, so I'll go over the answer pretty quickly then. So here, if you look at the crossings, first of all, if you try to follow it with your left hand, you'll see that you can trace around this part of the DNA. And as a result of that, you can see that you have two left-handed crossings right here. And as I mentioned before, if you have DNA in the plectonemic form that's left-handed, then that means when you're going to the solenoid form, it's actually right-handed. So this means that you're looking at DNA that it's left-handed in the plectonemic form, but when you go to the solenoid form, it's right-handed. And if it's right-handed in the solenoid form, remember that positive and negative supercoiling is defined from the perspective of solenoid form. So a right-handed DNA in solenoid form is positive supercoiling, which is the answer to this question. So those are all the slides for the first part of DNA supercoiling. Does anyone have any questions? Okay, so then I'll post these slides later and I'll hand it off to the next tutor.
Okay, so I'll be going over part 2 of DNA supercoiling. And one concept I want to start with is the definition of linking number. And so that's going to be the number of times one strand of DNA wraps around the other. Also define it as the total number of times the DNA strands cross each other locally and globally. So I'll write that here. And from this definition, we can see, we can get an equation for the linking number. So that's going to be L equals W plus T, with W is rive, and that's going to be the number of times the global axis crosses itself. So this accounts for the global, and then twist, and this will be the number of turns of DNA about the local axis. So if we want to visualize what the linking number will be for a short piece of DNA, I've drawn one over. And so let's choose a reference point. I'm going to choose. So here we're going to start, and then this will be one turn, linking number will be 2, and then this will be another turn, linking number will be 3. So for this short piece of DNA, L will be 3. Okay, and I've drawn a relaxed circular piece of DNA here, and for relaxed DNA, rive is going to be 0. And so from our equation, we can see that L is going to equal T. Linking number is not going to change for a closed unless you could with, for example, a topoisomerase, then you can change the linking number. And linking number also won't change if the ends are fixed. So this would be for a linear piece of DNA. Okay, does anyone have questions about any of that? Okay, so I'm going to go over positive and negative supercoiling. And so supercoiling can be positive or negative depending on the direction that the DNA is twisted. So I've redrawn my circular DNA, and let's say it has a linking number of 10, twist of 10, and the rod is 0 because it's relaxed. And so let's say we cut it right here, and we're going to rotate one end by 3 left-handed turns. So we're going to underwind it because the DNA is right-handed, and then we're going to rejoin the ends. And what we're going to get is, if we just rejoin them like that, there's this bubble in the DNA, but it will be underwound. And because we cut the DNA, that means that we changed the linking number. And so we removed 3 turns. So now the linking number is going to be 7. And the twist is also 0 because there's no supercoiling in this picture. But the DNA isn't going to like this conformation because it likes that optimal 36-degree rotation between bases. So the base pairs are going to rotate this. And what you end up getting is you end up getting this, you can't get this plectonemic form with 3 superhelical turns. And like Poojan said, they're going to be 3 supercoils with a right-handed sense of crossing. So if you, so my hand, if you put your thumb in the direction of the strand on top, so this strand, and then you curl your fingers around, right? You can see that it's going this way, like this. So that's a right-handed crossing. With 3 superhelical turns. So now linking number still is we didn't cut the DNA in any way, but the twist is now going to be 10 because it reverted back to its relaxed state, and the ride is going to be -3. And it's negative because we underwound the DNA. So the crossing is going to look like this. If I just draw a little illustration here. And this is in equilibrium with the solenoid form. So here's the solenoid form, and it's going handed. So if we see the direction that the twists are moving, it's going this way. And so we can, sorry, left hand. We can put our thumb in the, in the direction of this, and then if we curl our fingers around, we can see that it's curling with a left-handed sense for the solenoid form. Okay, so we're going to do the same thing, but this time we're going to cut the DNA and then rotate by 3 right-handed turns, so we're overwinding it, and we're going to rejoin the ends. And now we're going to get supercoiling that looks like this. So linking number is going to be 13 since we added 3 turns. Twist is going to be 10 because it's relaxed back, and then ride is going to be a +3. This time the sense of supercoiling is going to be left-handed. So here's our hand again, and then twist in this direction. And then for the solenoid form, again, is going to be the opposite. So the axis is now going this way. And then if we put our thumb in the direction of the axis and curl our fingers towards how the DNA is moving, we can see that it's right-handed. Okay, and this is left-handed. Okay, any questions about that? Okay, so now I'm going to talk about how DNA replication induces negative supercoiling. And this happens because the polymerase is fixed because it can't rotate. And this is because the polymerase is pretty big, so it's not going to rotate with the DNA as it unwinds. And then, so one end is fixed here, and the other end is going to be fixed over here. And this can happen in the cell for a lot of reasons, like histones, or if the chromosome is really long, you can basically say it's fixed at the end. So what happens is the polymerase is going along and it's adding new nucleotides without rotation because the polymerase itself can't rotate. So this is what I've drawn here. This is also in the lecture slides. And linking number is going to be constant because both ends are fixed. And so the length of the DNA is increasing as new nucleotides are added. So this means the twist is going to increase because the base pairs will relax to their optimal rotation, and from our equation, writhe must decrease. Linking number equals twist plus writhe. So if this increases and L is constant, then that means that writhe has to decrease. And so here, if we have the polymerase starting and it's synthesizing this double-stranded DNA, and then it adds some base pairs here. So let's say that it adds 1 turn's worth of base pairs and then it relaxes, then we're going to get a negative supercoil behind the polymerase. Any questions?
I kind of have one question. Can you hear
Yeah.
me? So is it that
Yes.
Is it that the, like, I'm trying to like think of like the order of it. Is it like it twists and then like it, I don't know if there's an order if it happens at once. I don't know if you can maybe explain. Like as
Yeah, I think it would happen all at once because as the base pairs settle into their preferred at the same time, it's going to force the DNA back into the supercoiling. And if it didn't happen at the same time, what would happen first is, like, let's say the base pairs are twisting back 36-degree rotation, but then there's going to be a lot of tension in the DNA. And so that tension is going to force the DNA to recoil pretty soon after that if they are sequential. Yeah. Okay. I wrote a question on the worksheet and I'll just read it here. So you find a DNA polymerase in the process of DNA replication. Assume initial conditions where the DNA polymerase is about to add to a relaxed DNA strand with a length of 1,000 base pairs. I'll write that here. And if the polymerase adds 30 base pairs, calculate the writhe and twist after the DNA relaxes back to optimal rotation per base pair and assume 10 base pairs. Okay, so it told me the values of the writhe, twist, and linking number in its relaxed state.
In its relaxed state, would the twist be 10 and the linking number 10 and the writhe is 0?
Add one more 0 to your answers. So it's going to be 1,000, right? So 1,000 base
100,
pairs.
yeah, my
and
bad.
if you
my
have
bad,
10 base, yeah. Okay, yeah, good. And that's 1,000 ON. Yeah. Okay. Price adds 30 base pairs. Okay, and someone scream at the linking number.
Would it be 103 or 97? Because if the polymerase is adding 30 base pairs and there's 10 base pairs per turn, so you're adding 3 turns. So we're talking about linking number. The linking number will 103, I think. I don't know.
Okay, so you're on the right track with the twist. So the new twist
or the
is
linking
going to
number
be,
remains the same as 100. Okay.
Yeah, that's right. Yep.
And the twist will be 103.
So this is going to
and
be,
then
right.
the writhe will be -3.
Okay.
Yeah, exactly right. Yep, because we need these to all add up. So -3 equals 100. Okay, does that example make sense?
Do you know that the linking number is fixed because or it remains this constant when we're adding to the polymer or the polymerase when it's adding nucleotides because both ends are fixed of the DNA. Okay.
Yeah.
I got it.
Exactly right. Yep. Because polymerase fixed, and then somewhere in the cell, you're going to have the other end of the DNA fixed.
Okay.
Okay. Okay. Yeah. The next tutor may go.
Okay, hello everyone. Just let me share my screen real quick. Can everyone see my screen okay? Let me rotate this. So I'm going to be talking about non-standard base pairing and RNA structure. So before I get into like the nitty-gritty details, I think it's kind of like good to set context, right? Because I feel like we all learn about DNA first, and it's kind of this implicit assumption that Watson-Crick base pairing or standard canonical base pairing, which is, they all mean the same thing, is good, and non-standard base pairing is bad, right? Because if you add a wrong base to your DNA template, then you don't properly replicate. And that's obviously not good for the cell. But I think it's important to understand that non-standard base pairing is really important and very common in RNA. So why is that? Well, for DNA, the whole purpose of DNA is to have accurate replication, right? You have this genome and you want to give it to your daughter cell, and you don't want to screw it up so badly that your daughter cell is messed up. But for RNA has a lot of different functions. This is not in your class, but I think at least most of you will have seen some of it in your biology classes, right? So we have tRNA, mRNA all involved in transcription. We have ribosomal RNA involved in translation, snRNA involved in splicing mechanisms. So there's a lot of different things that RNA is responsible for. So if we constrain RNA to the same structure and functions as DNA, it can't do as many things. So how is it that RNA is able to accomplish so many different functions when it's basically the same, almost the same for nucleotides minus the, 2' hydroxyl. So RNA can do this because it has non-standard base pairing, right? And it's single-stranded. So that allows it to adopt conformations and shapes that are vastly different from DNA. And that allows it to assume a lot of different functions. So let's go through the first example of a non-standard base pair. Just want to say first that there are a lot of examples of non-standard base pairing. It's basically just anything that's not your Watson-Crick base pairing. So you should not in your studies be trying to memorize all the possible non-standard base pairing combinations because it's just not really efficient. I think the most important thing is to understand the concepts. When there is something that you should memorize, I'll point it out. So for your GU wobble, I've drawn it out here, right? So let's take a look and compare it to our standard Watson-Crick CG base pair. If you look at here, we have 3 hydrogen bonds. Can everyone see that? So you have 3 hydrogen bonds, and the extra cyclic amine right here is involved with is a hydrogen bond donor to the carbonyl oxygen over here. But if we look up here, where guanine is a hydrogen bond or base paired to uracil, the amine here sticks out. So you can already see that the presentation of functional groups is slightly off. And the shape too is a little bit different. So if you look at the bond angles with respect to the glycosidic bonds right here, for your standard base pairing, it's 54 degrees. But if you create a GU wobble, one of them is 45 degrees and the other is 65 degrees. So not only do you have a change in the presentation of functional groups, you have a change in the actual shape of the base pair, right? So this doesn't happen in DNA because when you have your polymerase, you have mechanisms that check the shape of your base pair. This is not, I don't think in your lecture notes, but I think it gives good context. But for RNA, because you can create these non-standard base pairs, you have a greater repertoire of structural variance. You have this amine, you have more exposure to these hydrogen bond donors, which allows you to do different things. For example, if you look at this picture, this is a picture from your textbook of tRNA. This is right here is your standard CG base pair. You can see the red is negative and the blue is positive, right? So you can see that because of the wobble here, the electrostatic surface looks different than the electrostatic surface here. The way the charges are distributed because of the non-standard base pairing is different. You also see that the shape is different. So here you have this crooked thing that's flipped over, and then in the wobble, it straightens out. I mean, this isn't something you should be able to predict, or you cannot predict just from like, oh, it's a GU wobble. But the fact is that different things happen because you have non-standard base pairing. And what this allows in biological systems is you can have increased specificity for certain proteins, certain DNA or RNA sequences. You have an affinity for different ligands. So for example, the GU wobble, because of the extra cyclic amine right here and the extra hydrogen bond donors up here, is particularly good at bonding divalent metals. You can read about it in your textbook. The details aren't really important, and you probably won't be tested on it, but it's just important to realize that because you have non-standard base pairing, it gives the base pair unique properties, allow you to do different things. So the GU base pair compared to other standard base pairs is also a lot more stable. So if we go down, I think we can just go into that later. So another example of non-standard base pairing that you should know is Hoogsteen base pairing. So this is an example of base pairing that you should memorize for your exam. It's come up in past exams. It came up in my exam. So just make sure that you are able to draw and identify Hoogsteen base pairs. So the definition that Professor Korean has in his textbook are that Hoogsteen base pairing is hydrogen bonds between Watson-Crick base pairing edges of one base and major groove edge of another base. So when I was learning this, that didn't really make a lot of sense to me. So the more precise or specific explicit definition of Hoogsteen base pairing from Hoogsteen is that Hoogsteen base pairs use the C6N7 face of the purine hydrogen of the purine for hydrogen bonding with the N3C4 face of the pyrimidine. So what does this mean? So let's take a look at the picture in Professor Korean's textbook. So here's the Watson-Crick base pairing, right? We can see the guanine and the cytosine. So this is your standard Watson-Crick base pairing. Here we have a cytosine. It's hydrogen bonded to the guanine, but this is not the way it's typically hydrogen bonded in, say, DNA. So how is it hydrogen bonded? Well, you have the pyrimidine here, right, in the definition, the N3C4 face of the pyrimidine. This is the N3C4 face of the pyrimidine. And then the definition, it hydrogen bonds with the C6N7 face of the purine. The purine is the one with 2 rings. So this is, I've labeled with numbers for you. This is the 6, 7. This is the 6, 7 face of the purine. So that's where the hydrogen bonding occurs. And the same thing happens if you have a different Hoogsteen interaction. Again, this is your AT standard Watson-Crick base pairing. And you have from the purine, the C6N7 face, hydrogen bonds with the N3C4 face of the pyrimidine right here. So that's how Hoogsteen base pairing is defined. And you should like try practicing drawing these things, recognizing it. And one thing that really confused me when I was learning it is that when you look it up online, instead of showing 3 bases as your textbook shows, sometimes they only draw 2 bases. But those 2 are actually just the same thing. The Hoogsteen base pairing interaction is just this component. It's just the side component. It's these 2. The extra Watson-Crick base pair is just to kind of illustrate how that happens in real biological systems, right? So why would having Hookean base pairing be useful? Why would you form an extra base pair with a double-stranded nucleic acid? Well, this actually occurs, for example, when you want to silence a certain gene. So Professor Kurian goes into an example in his textbook. I'm sure you don't have to know the particular example, but I think it illustrates a concept. So let's say you have so a regular gene and you don't have that third nucleic acid, right? Then you can have your whatever protein right here, call it transcription factor, some general thing. You can bind to the DNA and you can make mRNA and you can express your gene. Again, this isn't something explicitly taught in your class, but I think it gives context. But if you have a single-stranded RNA that can Hookean base pair with your double-stranded Watson-Crick, it can get in the way of proteins that would otherwise allow the gene to be expressed. So this kind of triplex formation is actually used in biological systems to regulate gene expression. And these other examples aren't particular. I'll post these notes, but these are just more examples of the same concepts. Yeah, for example, you have RNA stems and loops, which is, well, one, it's intramolecular hydrogen bonding, so you can form these unique structures that you wouldn't find in DNA. But here again, you can find the UG non-standard base pairing because it confers additional thermodynamic stability and other properties that allow RNA to have different functions. So I think I'm running out of time, so I won't go on any further. But does anyone have any questions or any part of my handwriting they'd like me to clarify? I know it's a little bit hard to read.
I have a question for the Hookean base pairing.
Yeah.
It doesn't have to be between one purine and one pyrimidine, right? It's just like if it's involved in that pairing, then it has to use those phases.
In the textbook, the technical definition, if you look at what Hookean wrote, because this is something I had trouble with too. It's between a pyrimidine and a purine, and in Professor Kurian's textbook, those are the examples he's given. So I would stick with knowing those. In practice, maybe other people used other definitions for Hookean base pairing. You might have a let's say purine bonded with a purine in a Watson-Crick base pairing, but I haven't seen that.
Okay.
Sorry if that's not really helpful.
I just think we'll get to it later. I think two purines are Hookean base pairing in the homework, but thank you so much for that. Yeah.
Okay. Does anyone else have any questions? Okay, if not, I will just hand it back to Pujan.
Awesome, thank you. All right, so that concludes our Wednesday concept review session. I'm going to stop the recording